next week's paper, which I hope you guys will love. I mean, prions are really crazy things, and so hopefully it's at least novel and interesting to talk about if the paper is not good. A new mechanism for transmissible prion diseases. So I just want to do, in case people haven't heard about prions yet, briefly, what are prions? What are the diseases? What are the structures of the proteins that get formed? And then a little bit about approaches. We all have the prion protein gene. We do not all have prion protein disorders, hopefully. Normal-ish gene. It's located on chromosome 20. It makes a 253 amino acid protein. The gene name is PRNP, prion protein. Okay. Have no idea what it does. Why do we have this gene? Don't know. There's a lot of opinion. There's a lot of people that are interested in figuring out what it does. It may be a membrane-bound protein. It might be involved in signal transduction. Maybe. So again, we have this protein, or what it normally does for us. There's a lot of terminology in this paper, as with a lot of papers, but this one's particularly heavy in it. So this is why this figure is here. And remember, these go on Blackboard, the slides and the actual audio as well. So if you want just a PDF of these, you can refer back to these when you're reading the paper if you want. So this paper uses all of these different acronyms. PRP, prion protein, is the same thing as PRPC, prion protein cellular, or the normal version of the protein. Then we've got the SC version of prion protein, or scrapie, which is the name of the sheep disease, sheep protein disease. Because when sheep get scrapie, apparently they do this sort of a thing a lot on the sides of their pen. So they call the disease that sheep get scrapie, but it's a prion disorder. That's what I heard. Then there's rPRP, recombinant prion protein. That is, they make it in vitro in a dish or in a test tube. So it's been cloned, it's a gene that's been cloned out of an organism. They can make in vitro protein, not proteins that have been taken from people. In fact, this lab makes a point later in the paper that their lab has never actually worked on the mutant version of the prion protein. So they don't touch versions of prion proteins that could give them prion diseases, essentially. So these were recombinant prion protein. And then there's the version that they make in this paper, PRP-res, prion protein resistant. And resistant means resistant to proteinase K digestion, and we'll talk about that in a second. Prions are the things that break a lot of rules in the central dogma, which is why they're so cool. Right? DNA makes RNA makes protein. What's the transmissible thing? What's the transmissible molecule? What transmits information from generation to generation? What do we inherit? It's DNA, genes, etc. So for eons, since the time of Darwin, not eons, right, geneticists have made this big point about the only thing that's transmitted is DNA. But then comes proteins that can also transmit disease from generation to generation. So then we're not talking about DNA being the only molecule of heredity anymore, proteins can too. This figure starts the brief conversation about how that works. So on the left is the conformation of the normal prion protein, PRPC, the cellular version. It's mostly alpha helical. What happens in these prion diseases is that protein, same amino acids, changes structure somehow, don't know how, and adopts this highly beta-pleated sheet version of the same protein, just different fold on the right. So the essence of prion disorders is normal protein becomes mutant, misfolded protein. Same amino acids, different structure. The one on the right does bad things to us. The one on the left is fine. We've got the gene, we make the protein, it's normal, we're okay. But when it misfolds, really bad stuff happens. So how can a protein be a transmissible agent? I can give one molecule of prion protein to somebody and that can give them the disease. This is how that works. in cartoon form. This is how it's thought that this works. That you start out with the cellular, the normal version of the protein, and somehow something makes it happen that that protein misfolds into the bad beta-pleated sheet structure. It might be by chance, there might be certain conditions that cause that misfolding to occur, and we don't know yet. But when you get a misfolded version of the protein, this is the wacky but highly supported theory. That misfolded protein catalyzes the misfolding of the normal version into more of the misfolded version. That's the transmissible part. You get one molecule and that effect amplifies. So one misfolded protein is horrible. Even just one misfolded protein, because that starts the process of converting all of the normal prion protein into all misfolded prion protein. And then supposedly those masses of protein clump together and cause amyloid plaques. So in your neurons, you get these huge aggregates, bodies of protein that mess up the neurons, and then you get neurodegeneration, neuron death, then really bad things. It's very similar to a hypothesis of how, I think, is that Huntington's? No, it's not Huntington's disease. Alpha-synuclein. It's one of the other famous neurodegenerative disorders. Parkinson's? It might be, yeah, I think you're right. Thank you. Where this other protein does basically the same thing, it's thought. That it misfolds, and then it causes these nuclear inclusions, plaques, that are called Lewy bodies in that disease. And that also causes neurodegeneration. There are lots of different neurodegenerative disorders that supposedly, that scientists think work along the same lines. That protein misfolding causes neurons to die, and that causes neurodegeneration. So prion proteins doing this is not unheard of, that they might cause misfolding of other proteins, but it's still a strange thing to happen. A single protein can cause other proteins to misfold, and that can cause some serious issues. So in terms of prion disorders, this is where it gets really weird. There are 3 different types. You can inherit prion disorders. There are heritable prion disorders. Then there's sporadic and there's acquired. So we're going to go just really quickly through what I mean by each. Okay. So when you've got a heritable mutation, that's actually talking about DNA transmission. So you inherit from your father or your mother a mutation that predisposes your prion protein to misfold. So there are a number of genetic mutations that can lead to higher risk of having a prion disorder. Those are the heritable ones. Sporadic happens all the time. We think that most of the human cases, or at least a significant number of the human cases, are sporadic. That thing what I said, you know that thing what I said. That just by chance, one of your prion proteins misfolds. And that starts the process of neurodegeneration. So it can happen at random to anybody, anytime, at a very, 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 very low frequency, but that would be sporadic. It just appears out of nowhere. Then there's the one that everybody knows about, which is acquired, or when you get the protein, not a DNA mutation that predisposes you, not that your protein misfolds on its own, but you get a misfolded protein in your body, in your brain, somehow. So you acquire it. We're getting there. So Creutzfeldt-Jakob is one of the many human prion disorders. The one you've never heard of before, except Ben mentioned it a couple weeks ago, I think, is fatal familial insomnia, one of my favorite genetic disorders. That's one of the heritable. You get a mutation from a relative, and this is a prion disorder, and basically you do what it sounds like. You, at a certain age, like 20 or 30, you sleep less and less and less, and then you never sleep at all, and then you die. You stay awake to death, literally. Then there's Kuru, the one that almost everybody has heard of before in humans, which was, I think it's Papua New Guineans, someone in Micronesia or Southeast Asia, who the culture was that you consume your dead relatives, and then It's really easy to, because you're eating proteins, if one of those individuals has prion proteins in them, then you ingest them, then they might stay in you, and then they might do bad things to your prion proteins. And so, since that practice has been essentially discontinued because of this issue, it's not really an issue anymore for them. Then there's non-human versions, right? Mad cow disease is great. So mad cow is the cow version. Cows have prion proteins too. And the big problem with cows, which you've probably heard of, because especially in England, they're really concerned about mad cow disease. And if they get even the hint that one of their cows has a problem, they euthanize it, burn it. They kill whole herds of cows when one of them gets it, just in case it's passed it on to some of its neighbors. And one of the reasons for that, why cows and sheep particularly are affected, is because of the way that feed has been generated for those animals. When you wind up giving cows cow parts in their feed to save money, that's how protein transmission can happen from cow to cow, when you've got essentially cow cannibalism based on what you're feeding your cows. So when you stop doing that, then it turns out that transmission of mad cow disease isn't as big of a deal. It still can happen, but it's much less likely. So basic rule of thumb, don't eat your own species. I already talked about this briefly. A bunch of different, as I mentioned, neurodegenerative disorders all have the same sort of effect. The proteins aggregate in neurons, nuclei of neurons, and that somehow that causes neurodegeneration. So Lou Gehrig's disease, Parkinson's, Huntington's disease, Alzheimer's, etc. So prion diseases are not unique in this regard. They're a type of disease that has a lot of different diseases of that type. One of the reasons that prion protein diseases are hard to manage is because it can take a really long time to manifest symptoms. So you can be symptom-free but able to transmit for a long period of time. Right? So often in humans you don't get diseases until you're 40, 50, 60, if it's one of the inherited type. If you acquire it, then you can acquire it at any point in your life. So this is one of the problems, is that it takes a while for those few bad proteins to convert more of the bad proteins and eventually cause problems in the neurons. So getting to the paper, one of the basic ideas in prion biology for a while is that there's a specific structure of prion protein, one specific conformation that it folds into that makes aggregates. And it's the structure and shape of those aggregates, multiple copies of these misfolded prion proteins, that are essential to catalyze the misfolding of other proteins. So it's not just that it misfolds, it has to misfold into a particular shape. And these authors are saying that they found a new shape that nobody knew about before that they say can cause prion diseases. And this might be a bad thing, because if we're only focusing on testing for and detecting certain misfolded prions when we do things like testing cows for mad cow and testing sheep for scrapie and testing humans, we might be missing detection of a whole host of, or at least one, additional type of protein fold that might be transmitting to other people that we're not detecting or we don't know about yet because of how long it takes for prion disorders to become apparent. Okay. One of the so now we're down to techniques, just a few more bits of information. There are 3 different antibodies that they're going to use in this paper, so I just wanted to point them out. They're called 3F4, D18, and SAF84. Those 3 antibodies recognize 3 different regions of the prion protein. And so what you can do with these is ask which regions, that is amino acids. So amino acids 109 to 112, 133 to 157, and so on. Are those regions folded in the structure that those antibodies recognize, or are they folded in a new way that those antibodies can't recognize? So remember, if you have a 3D structure of a protein, an antibody recognizes a specific shape of that specific part of the protein. So this might be 3F4, and that might be residues 109, 110, 111, 112, the 4 different amino acids in that part of the protein that it's recognizing. And so you could have an antibody over here that's D18, and you might have an antibody that recognizes that part, SAP84. So if you want to know if this protein is folded in a new shape, like that, what happens when you try to detect this protein with these 3 antibodies? Depends on whether or not it's folded the same or not. So maybe this loop is still that loop, so maybe you can still detect this new protein fold with the same antibody. But maybe this part of the protein that SAP84 used to recognize is now in the middle, so you don't detect it anymore with that antibody. So this is what's called epitope mapping.
So it depends on the folding?
Right. So you can detect whether a protein has changed its structure by asking whether or not at one point you could detect that protein with one antibody and later you can't. The protein's still there, you should be able to detect it, but if you no longer do, that suggests the protein has changed its structure. So the antibody doesn't recognize that specific region of the protein anymore. So that's epitope mapping. Okay. The most important approach that they're using, which is really cool, and I have no idea if it actually works the way they say it does, like I can say anything. Serial PMCA, protein misfolding by cyclic amplification. This is a picture of serial PMCA, but basically it's PCR for proteins. So the question is, prion disorders are really tricky because you can have a little tiny fraction of your total prion protein population be misfolded. And how would you detect 1 or 2 copies of protein that are misfolded in a whole bunch of prion protein? So, and this is ridiculous, and in a really interesting way. They've made a mechanism by which they can make more misfolded prion protein. They're making like prion central. It's like terrorism. They've come up with a way to turn one misfolded prion protein into millions of copies of misfolded prion protein. Hope terrorists don't get their hand on this and put it all in our food supply. But what they do is they take a bunch of, so this is what happens in our bodies, they just try to speed it up. They want to have a mechanism where in the lab they can detect a single copy by doing this. So you take a bunch of C, prion protein C, the normal cellular type, you throw in 1 molecule of this misfolded, highly beta sheet, right? Not alpha helical, but beta sheet, prion protein scrapie form, the infectious form. And over time, that will presumably, according to this theory, cause those to misfold. But it takes a really long time. So what you can do is you sonicate that protein, you break it into little bits. And supposedly then those little bits of this misfolded prion protein cause more of those normal versions of the prion protein to misfold faster. And so that's why they call this amplification of converting units. You make more misfolded protein, you sonicate it again, that breaks apart those beta sheets into new multiple versions of the prion protein that go forth, and this is why the amplification part. So over time, you keep sonicating, you keep growing new beta-pleated versions, you keep sonicating, making more of these infectious, they're nucleating, basically, the conversion of more of this protein and more of this protein. That's kind of like why it's PCR-like. Every round of this, you amplify the amount of prion protein that's misfolded that you make, and eventually you get enough protein in your test tube that you can actually do a Western blot or epitope mapping and find out whether or not the structure of the protein in the tube after PMCA is the structure you expect, a new fold of the prion protein, the misfolded version that we already knew about, etc.
So it doesn't convert them into the misfolded version, it just converts them, so right here, prion with the 2 different conformations.
It it takes normal prion proteins, the wild type versions, and converts them into some other structure.
So not necessarily the misfolded version.
It's, yes, so it supposedly is converting them into the same structure as the infectious version. So you take a lot, so the way this starts, just like PCR, you take a lot of building blocks, that is the normal protein, lots and lots and lots of PRPC. You just throw in a tiny bit of the infectious form. And what they're trying to do then is make that infectious form convert the normal into infectious form, just do it faster than it would happen in a body, in an individual. To do it in a tube, and to use sonication to try to, because what does sonication do? When you blast proteins with sound, it breaks them, it also causes them to denature. So I think what they're basically trying to do is unfold the proteins, refold the proteins, unfold the proteins, refold the proteins. And if they do that enough times, which you do by sonication, they sonicate, they wait, they sonicate, they wait, they sonicate, they wait. The more time you unfold and misfold the proteins in the presence of a tiny little bit of the infectious form, that's supposed to convert all those, according to this theory, that's supposed to cause those wild type versions to convert into the infectious structure. Right? That's why I said this is wild. Does it work like they say it does?
Sounds like zombies for prions.
Yeah, sort of, yeah. Brains. Yeah.
Instead of a thermostator, a sonicator.
Yeah, you've got like a tube and a sonicator. Yeah.
Are all the misfoldings, do they have the same configuration or are they different?
We'll find out. So that's one of the so that's one of the questions of the paper is, does the structure matter?
And when we throw in an infectious one into the normal one, will it change its conformation into its conformation or some other misfolding? Will it change?
Don't know. Supposedly, I mean, proteins can change their structure all the time. Just by chance, they're a little bit malleable. You might change structure. So I guess the idea is the prion protein, the bad conformation is more stable maybe, so once the proteins fold in that structure that they don't change back to the normal wild type version. Problems with proteins, or problems with prions rather. A lot of prion proteins fold in such a way that, this is the really crazy part. Proteinase K, a protein that digests proteins, degrades proteins, cleaves amide bonds. That's its job. It's a protein that digests other proteins. Can't digest, can't degrade prion proteins in their scrapey form. That's one of the really bad things about the scrapey conformation, that they're really resistant to being degraded, destroyed. So we can use this to identify whether or not we've got prion proteins in their cellular conformation. So if you've got a bunch of protein, you throw a bunch of proteinase K on, if the protein disappears, you knew it was the normal protein. If it sticks around, then you know it's prion protein. That's the scrapey conformation. So that's another approach to the. Another one is related, guanidinium hydrochloride stability. So these prion proteins in the scrapey conformation are not only resistant to protease, but they're also really, really, really stable in this molecule, in this chemical, guanidinium hydrochloride, which is one of the strongest protein denaturants that's known. And so if the protein can stay folded in high concentrations of this, that suggests that it's the prion protein, the scrapey conformation. Okay, so they use this as well, so that you can ask, what does the protein look like in different concentrations of guanidinium hydrochloride? If it stays the same in low concentration, medium concentration, high concentration, super high concentration, it's probably the scrapey conformation, not the wild type conformation. Okay. So I'll end with the scary stuff. Prion protein in the scrapey conformation is really, really hard to sterilize. Can't digest it with proteinase K. You can't denature it with chemicals. You can heat it, but it's also resistant to heat. So you can't cook your burgers well enough to kill mad cow prions because the protein is resistant to denaturation by heat. You cook your beef all you want, but if it's got prion protein in the scrapie conformation, it's going to stay that way, most likely. And radiation. So what are we going to do if you get this sort of protein structure in your food?
Smash it.
Something, yeah, I mean, what are you going to do?
They're resistant to pH.
And it's also resistant to changes in pH.
Oh my god.
To a certain extent. I mean, nothing's totally invulnerable, but you should see, I didn't put in here, I toyed with it for a second, the CDC guidelines for how to sterilize medical instruments to ensure that, like, I've had brain surgery. I was hoping that they weren't using tools that they had used to do some surgery on somebody who had a prion disease and didn't know it. So the CDC has guidelines for how to sterilize all medical equipment to make sure that you don't pass unknowingly prion proteins from patient to patient. And it's something like soak the instruments in 5 normal sodium hydroxide for 3 hours and then rinse them and then autoclave them and then soak them in an ionic detergent for 7 hours and then rinse them and autoclave them and then do something else for 3 hours and autoclave them. It's like 4 autoclavings in 3 different solutions. You soak the metal instruments in before you give them the last sterilization and use the instruments again. So maybe that sort of thing would work, but you don't want to do that to your hamburger either. So that's the last question really is, in the big picture, is how do we control prion disease? And that's kind of what this paper is about. Identifying and then starting a conversation about what are we going to do about prions.